Solving the cholesterol conundrum. Hamburgers are a staple in the American diet, making it no surprise that heart disease is the #1 killer. But what is it about this delicious treat that makes it so deadly? The answer is cholesterol. Foods high in saturated fats, like hamburgers, are packed with cholesterol. Dietary cholesterol adds to the already large portion of cholesterol that is synthesized in the body. The liver is a cholesterol powerhouse, creating 75% of the cholesterol that ends up in your blood. When too much cholesterol enters the blood, dangerous plaques in the arterial wall can form and eventually lead to heart disease. The solution to high cholesterol is not simple. Scientists have long known that arterial plaque causing heart disease is formed from excess cholesterol in the blood. What they didn't know was how the interaction between cells and blood cholesterol was leading to the accumulation of dangerous plaque, and how to prevent the plaque from forming. In the 1970s, scientists Michael Brown and Joseph Goldstein determined that cholesterol is transported into cells using a surface receptor, and that the receptor ultimately mediates feedback control of cholesterol synthesis. Goldstein and Brown created this clear picture of cholesterol metabolism and cholesterol transportation while working with patients suffering from FH, familial hypercholesterolemia. 1 in 1 million individuals are born homozygous FH, which is the most severe form of the disease. Those who are homozygous FH have extremely high concentrations of blood cholesterol and suffer from heart-related diseases early on in life. Patients with FH have abnormally high levels of blood cholesterol. Such levels could not simply be explained by high cholesterol diets. Something was preventing the removal of cholesterol from their blood. Goldstein and Brown found that something to be a mutation in their Ldl receptor gene. The mutation caused defects in the Ldl receptor. The defective Ldl receptor prevented proper cholesterol removal from the blood, causing cholesterol to accumulate in the blood and eventually form plaques. Arterial plaque is made up of low-density lipoproteins, or LDL. When cholesterol levels in the blood are high, LDL gets trapped in the artery and accumulates in the artery wall. Lipoproteins are the packages that cholesterol travel through the blood in. The packaged cholesterol is encased within the hydrophobic core of the molecule. Lipoproteins are a combination of lipid and protein that allow for water-insoluble cholesterol to easily make its way through water-based blood. The two main types of lipoproteins when discussing heart disease are LDL and HDL. LDL transports cholesterol from the liver, where it is synthesized, to other cells in the body. Cholesterol is needed in these cells to form cell membranes, make bile acids, and produce hormones. HDL does the reverse of LDL. HDL collects excess cholesterol from the cells and takes it back to the liver, where the collected cholesterol is then recycled. Although LDL is hydrophilic enough to travel through the blood, it is too hydrophobic to pass through cell membranes, creating a need for something outside of the membrane to allow for their transport in. Lipoprotein receptors, known as Ldl receptors, allow for the entry of LDL into the cell. LDL cholesterol uptake functions via receptor-mediated endocytosis. LDL circulating in the blood binds to the Ldl receptor, and once bound to the receptor is engulfed into the cell. When the LDL enters the cell, the Ldl receptors are still attached. The receptors in LDL are later separated in the endosome. Once separated, the receptors are recycled back to the cell surface, where they will be used again in the cholesterol uptake process. The naked LDL is then delivered to the lysosome, where it is broken down, allowing amino acids and free cholesterol to be released. The newly free cholesterol is sensed by the cell. If there is too little cholesterol inside the cell signals for an increase in Ldl receptor transcription, which allows for more surface receptors to form, and as a result, an increase in LDL uptake. When there is excess cholesterol in the cell, 3 feedback mechanisms occur. The overall cholesterol synthesis is suppressed in the cell. Then ACAT is activated, allowing excess cholesterol to be stored in the cytoplasm as cholesterol droplets. And the LDL receptor synthesis is inhibited, limiting the number of surface receptors. Because patients with FH have a mutated Ldl receptor gene, the receptor's function is disrupted, and the receptor is ultimately defective. A defective receptor is unable to optimally collect and intake LDL from the blood. Poor LDL uptake causes excess LDL to accumulate in the arterial wall and eventually leads to heart disease at an early age. Individuals with a non-mutated Ldl receptor gene have normally functioning LDL receptors. In such cases, hypercholesterolemia is a result of too much dietary cholesterol. With more cholesterol entering the blood from diet, there's too much cholesterol for the receptors to take in, leaving excess cholesterol in the arterial wall. In conventional hypercholesterolemia, heart problems are experienced later on in life. The discovery of the Ldl receptor by Goldstein and Brown answered the question of how LDL was taken from the blood into cells and proved to be the missing link to understanding cholesterol regulation. But the question of how to treat and prevent hypercholesterolemia still remained. Around the same time in the 1970s, scientist Akira Endo discovered statins, a class of compounds that lower LDL cholesterol levels in the blood. Previous cholesterol-lowering agents available were neither effective nor ideal for treating high cholesterol. Endo's discovery of statins stimulated the development of the effective cholesterol-lowering drugs lovastatin, atorvastatin, and fluvastatin. Endo decided to look for a cholesterol-lowering agent in fungi. Endo tested 6,000 microbial strains of fungi in a cultured broth to see which would prevent cholesterol production. Through trial and error, Endo discovered 2 compounds that lowered cholesterol levels. Citrinin was the first substance Endo found that inhibited the production of cholesterol. However, citrinin was found to be toxic to the kidneys and was dropped as a potential treatment option. The second compound, compactin, was found to lower LDL cholesterol just like citrinin, but without negative side effects. Compactin effectively lowered LDL cholesterol without harming the kidneys, making it the first safe statin to be discovered. But how exactly do statins inhibit production and reduce cholesterol levels? Cellular cholesterol production takes place in the ER of cells, where the molecule HMG-CoA is found. Cholesterol is synthesized from HMG-CoA through a series of cascading reactions. HMG-CoA reductase is a critical enzyme in the cholesterol synthesis pathway, as without it, HMG-CoA cannot be reduced to mevalonate and the pathway cannot progress. In order for HMG-CoA to activate the cholesterol synthesis pathway, the molecule must bind to the enzyme HMG-CoA reductase. By binding to HMG-CoA reductase, cholesterol synthesis can occur. Statins are structurally similar to HMG-CoA and can also bind to HMG-CoA reductase. Statins' similar properties to HMG-CoA allow it to compete for the reductase binding site. When statins are present, they prevent HMG-CoA from binding to the enzyme by binding to it first. With the reductase unavailable to HMG-CoA, the molecule cannot be converted to mevalonate, and the cholesterol synthesis pathway is halted. In this way, statins prevent cholesterol production by inhibiting HMG-CoA reductase. The inhibition of HMG-CoA reductase leads to a decrease in the amount of cholesterol produced in the liver. Because synthesis of cholesterol is reduced, the amount of cholesterol packaged as LDL is also decreased. The reduction of cholesterol levels in the cells causes an upregulation of the Ldl receptor gene, which in turn leads to an increase in the number of Ldl receptors. With more receptors on the cell surface, the amount of LDL taken into the cell is also increased. With more LDL being taken into the cell and less LDL entering the blood from the liver, the overall level of blood cholesterol is decreased. Statins' ability to lower blood cholesterol levels make them a potential tool for treating patients with less severe cases of familial hypercholesterolemia. Not all FH patients have the same degree of deficiency in their LDL receptor gene. Those that are homozygous FH have 2 copies of the FH gene and are extremely rare in severe cases of FH. These patients have a complete deficiency in LDL receptors, and because statins lower cholesterol by upregulating the receptor gene, they do not work in those that are homozygous. Those that are heterozygous FH only have 1 copy of the FH gene and thus have fewer defective receptors. In these less severe cases, high doses of statins do work, as there are more functioning receptors that statins can upregulate. Statins are not only used to treat FH patients, but are also vital tools for treating conventional hypercholesterolemia. More than 35 million Americans currently suffer from high cholesterol and are at risk for heart disease. 2013 guidelines put out by the American Heart Association and the American College of Cardiology revised the standards for heart disease treatment, making it that 56 million Americans between the ages of 40 and 75 years old would be eligible for statins. With an increasing number of Americans being diagnosed with high cholesterol, it is more likely that you, or someone you know, will be taking a statin. This life-saving treatment would not have been possible without both basic and clinical research. The work of Goldstein and Brown provided a better understanding of the cause of high blood cholesterol. This knowledge allowed for the statins that Endo discovered to be further developed into the cholesterol-lowering drugs prescribed today. Together, they solved the cholesterol conundrum.